Hi and Assalamualaikum everyone. So today I will be presenting about pollination and fertilization processes and the life cycle of flowering plant. So before we dive into this subtopic, I would like to introduce myself. My name is Raja Nushazwani Shamira binti Raja Effendi. My student ID is 2025236066 and I am from class M3802402C. So here are the table of content about this subtopic that I will be explaining to you guys. So firstly, I will talk about the introduction of flowering plant, reproduction, pollination, types of pollination, pollinators, microsporogenesis for male gametophyte development, megasporogenesis for female gametophyte development, double fertilization, and lastly, plant life cycle. Let's go. Introduction. Flowering plants, also known as angiosperm, reproductive through sexual reproduction, which involve flowers as the main reproductive organ. The flower contains both the male and female structures. The male part is the stamen, which includes the anther and filament, while the female part is the carpel, which is consisting of the stigma, style, and ovary. By understanding these processes is important because they explain how plants form fruits, seeds, and the next generation of plants. Pollination. So what is pollination? Pollination is the transfer of pollen grains from the anther to the stigma. This is the first and most important step before fertilization can occur. Pollen grains are produced in the anther and contain the male gametes. Pollinators ensures the pollen reaches the female parts of the flower so that the male gamete can later fuse with the female gamete inside the ovule. Without pollination, fertilization cannot happen and the plant will not be able to form seeds and fruits. There are also two main types of pollinations. First, self-pollinations. This happens when pollen from a flower lands on the stigma on the same flower or another flower on the same plant. Self-pollination usually produces offsprings that are genetically similar to the parent plant. Second is the cross-pollination. This occurs when pollen from plant is transferred to the stigma of a different plant of the same species. Cross-pollination increases genetic variations and often results in healthier offspring. So next is pollinators. Pollination can happen through various agents known as pollinators. So here are the common pollinators, which is insects such as bees, butterflies, and flies. Wine, which carries lightweight pollen. Birds, for brightly colored flowers. Water in aquatic plants. And lastly, animal like bats and small mammals. Pollinators play a major role in ensuring successful pollination, especially in plants that depend heavily on cross-pollination. So now let's go dive into the microsporogenesis, which is the development of the male gametophytes. This occurs inside the anther. The microspore mother cell undergoes meiosis, which producing 4 haploid microspores. These microspores then develop into mature pollen grain. So each pollen grain consists of a tube cell which form the pollen tube and then a generative cell which divides to form 2 sperm cells. These sperm cells are required during the fertilization process. Furthermore, is about megasporogenesis for female gametophyte development. Megasporogenesis takes place in the ovule inside the ovary. The megaspore mother cell undergoes meiosis, but only one megaspore survives. The megaspore develops into the embryo sac, also known as the female gametophytes. So the embryo sac contains one egg cell, two polar nuclei, two synergids, three antipodal cells. So these structures are essential during the fertilization process, especially in double fertilization. So here are the double fertilization. One of the most unique features of angiosperm is double fertilization. When a pollen grain lands on the stigma, it germinates and forms a pollen tube. So this tube grows down the style and enters the ovule. Inside the pollen tube, there are two sperm cells. Sperm cell 1 fuses with the egg cell to form the zygote, which develop into the embryo. Sperm cell 2 fuses with the two polar nuclei to form the endosperm, which is triploid. So the endosperm acts as the nutritive tissue, which providing food for the developing embryo. Then after the fertilization, the integuments of the ovule develop into the seed coat. Then the ovary develops into a fruit, and it walls becomes the pericarp. So the last part is the plant life cycle. So flowering plants undergo an alternative of generations consisting of sporophyte stage and the gametophyte stage. So for sporophyte stage is the adult plants which produces flowers. Flowers generate reproductive cells that lead to gametophytes. So for the gametophyte stage, it includes the pollen grain, which is the male gametophytes, and the embryo sac, which is the female gametophytes. So it means after the double fertilizations, the zygote develops into a new sporophytes, completing the cycle. So these life cycles ensure genetic diversity, adaptability, and the continuation of plant species.
Before the fertilization event can occur, the ovule has to undergo some changes. At present, the ovule contains one reproductive cell, known as the megaspore or mother cell. This cell is diploid and undergoes meiosis, producing 4 haploid megaspores. In the majority of species, 3 of these megaspores degenerate, leaving only one surviving megaspore. This surviving megaspore expands and undergoes 3 rounds of mitosis to produce 8 haploid nuclei. As the nuclei have not developed any individual division, they initially share the same cytoplasm. This complete structure is known as the embryo sac. Within the established embryo sac, cell walls begin to form between most of the nuclei. 3 cells named antipodal cells form opposite the opening of the ovule, known as the micropyle. Another 3 cells form above the micropyle. 2 of these are synergids, and the other is the egg cell. This leaves 2 nuclei in the center of the ovule. These central nuclei remain together in one large cell. It is the egg cell and this central nucleate cell which will eventually become part of the double fertilization event. In order for the double fertilization event to occur, the male gamete, the sperm, must travel from the anther to the embryo sac within the female reproductive organ. The pollen grain contains 2 main cells. A cell named the tube cell makes up the bulk of the pollen grain, and the sperm cell, which at this stage is known as the generative cell. To reach the embryo sac, a pollen grain must land on the stigma. Once landed, it begins to germinate. The tube cell forms a long structure down the style and into the ovary. The generative cell travels behind the tube cell nucleus. Once near the ovary, it divides by mitosis to produce 2 haploid sperm cells. The pollen tube reaches the micropyle and releases the sperm cells into the embryo sac. One of the 2 sperm cells fertilizes the egg cell. This produces a diploid zygote, which will become the embryo. The other sperm cell moves up and fuses with both of the central nuclei, forming a triploid cell. This unusual triploid cell develops into an endosperm and serves as the embryo's food supply during early development. It is only angiosperms, flowering plants, which have this double fertilization characteristic, where a diploid zygote and a triploid endosperm form. Gymnosperms, pines, tracheophytes, ferns, and non-tracheophytes, mosses, lack this double fertilization feature.
In conclusion, pollination and fertilization are essential processes that allow flowering plants to reproduce and maintain their populations. Pollination enables the transfer of male gametes to the female parts of the flower, while double fertilization ensures the formations of both the embryo and the endosperms. Together with the plant life cycle, these processes demonstrate the complexity and efficiency of reproductions in angiosperms. So that's all from me. Thank you for listening.